Developmental biology of the skin. Introduction. The skin is divided into 3 layers. Epidermis forms the barrier to outside world and is the locus of immune surveillance and activation to prevent and combat infections. Dermis provides the main structural substance, collagen, of skin, forms a main interface to vasculature and nervous system in the skin, and intimately interfaces with epidermis to coordinate skin function. Hypodermis, the area beneath the collagen-rich dermis that is characterized by subcutaneous adipose tissue with a role in energy balance, as well as recently defined roles in epidermis crosstalk and immune surveillance. Cells of the skin formed during development. Keratinocytes, central cells of the epidermis, and form intermediate filament keratins for structural resiliency. Fibroblasts, central cells of the dermis, secrete collagen, the substance for dermis. Appendages, hair follicles, eccrine sweat glands, apocrine glands, and nail unit. Melanocytes reside in epidermis and synthesize melanin to absorb and block the sun's UV light. Macrophages, neutrophils, and mast cells. Langerhans cells, immune cells in epidermis, and internalize and present antigens to initiate an immune response. Sensory neurons, monitoring touch, pressure, temperature, hair follicle movement. Arterioles and venules connecting to cardiovascular system. Lymph vessels return interstitial fluid to the circulatory system. Merkel cells live in the epidermis and sense touch. Immune cells develop elsewhere include T cells, B cells, dendritic cells. Central developmental signaling pathways. These are WNT ligands for important post-translational modifications. P63, a transcription factor and the central regulator of epithelial identity. SH ligands, very important to hair follicle formation and function. EATR, ectodisplacin A receptor, part of the tumor necrosis factor TNF receptor family and critically important for the development of appendages. Notch receptor, which initiates transcription and epidermal differentiation. BMP, bone morphogenic protein, ligands which regulate hair follicle cycling and growth. HOX, homeobox, transcription factor important in large-scale body patterning. This picture shows structures of the skin, makes clear the complexity required during development for formation of all elements. Structures of the skin. The outermost layer of the skin is the epidermis. The cell that composes the majority is the keratinocyte, with the structural protein keratin abundant in that cell. Keratinocytes are stacked in layers of increasingly more mature states until they are effectively ghosts of cells that serve only as a barrier and have little to no energy consumption. The basal lamina is a vital scaffolding structure that separates the epidermis from the dermis. The dermis provides structural support to the skin. It contains fibroblasts, which are the main producers of collagen and other extracellular matrix proteins that give strength and elasticity. In addition, the dermis houses blood vessels, lymphatic vessels, and nerves, all of which are critical for nutrition, immune surveillance, and sensation. We also find a variety of hematopoietic-derived cells here, such as mast cells, macrophages, and dendritic cells, that contribute to defense and inflammation. Beneath the dermis is the hypodermis, or subcutaneous tissue, which is composed largely of adipocytes. This layer cushions underlying structures, provides insulation, and serves as an energy reservoir. General summary of embryonic development. Human development through 40 weeks of gestation and subdivided into trimesters. The most complicated steps at the earliest times. The skin begins to form within first 2 weeks. Summary of important developmental stages of skin development. First trimester, transition from embryo to fetus. 2 weeks. Epidermis, gastrulation begins, ectoderm forms. Dermis, single-layer germinativum appears. 3 weeks. Epidermis, periderm forms, serving as a temporary protective layer. 8 weeks. Epidermis, intermediate layer forms, melanocytes become present. Dermis, a distinct boundary between epidermis and dermis is established. Appendages, dental placodes start forming, precursors to teeth. 9 to 10 weeks. Appendages, nail and hair follicle placodes form. Epidermis, Langerhans cells become present. 12 weeks. Epidermis, Merkel cells are present. Dermis, dermal epidermal junction and basement membrane mature. Appendages, sweat gland placodes form on volar palmar plantar sites. Second trimester. 13 weeks. Dermis, papillary dermis becomes distinct from reticular dermis. 24 weeks. Epidermis, periderm sloughs off. Dermis, elastic fibers become visible. Third trimester. 27 weeks. Advanced differentiation continues, skin more closely resembles postnatal skin. 40 weeks, birth. Fully developed skin, ready to function as a barrier and thermoregulator at birth. Neural crest development. Begins in the third week of fetal life. Forms at the junction of the neural plate and ectoderm during gastrulation. Neural plate folds to form the neural tube. Crest cells remain free in the mesoderm. Neural crest cells migrate dorsally and ventrally after neural tube closure. In the head, they migrate early before tube closure and contribute to dermal fibroblasts of face and anterior scalp. They secrete WNT1 ligand, which activates beta-catenin, important for signaling and cytoskeletal regulation. Clinical relevance: give rise to melanocytes, fibroblasts, and are implicated in certain diseases. Somite development. By the third week, mesoderm condenses into cuboidal somites lateral to the neural tube. Somites primarily form the axial skeleton and muscles. Dermal fibroblasts originate from the dorsolateral somite region, dermatomyotome. Early somite fibroblasts also contribute to skin development. Epidermis differentiation. The epidermis is a strong barrier, built from stratified keratinocytes. Keratinocytes produce keratins, intermediate filaments that not only provide structural support, but also regulate various cell functions. Each epidermal layer expresses specific keratin pairs, type I plus type II, making the layers well-defined. Unique developmental stages. Epidermis undergoes special embryonic stages not repeated later in life. First is periderm formation. Second is intermediate layer formation. And third is full maturation. Early development starts after gastrulation. The ectoderm becomes a single cell layer, the germinativum. Cuboidal, undifferentiated, mitotically active. 15 days. Periderm forms above germinativum. Appears as flattened cells with tight junctions and polarized cytoskeletal adhesions. 60 days. Intermediate layer forms between germinativum, basal, and periderm, outer. Likely through asymmetric cell division. One daughter cell stays as a basal stem cell. The other begins upward differentiation. Stratification and barrier formation. Once these three layers exist, stratification begins, transition to fetal skin. Barrier formation initiates at dorsum and head, spreading posteriorly and toward the midlines. Controlled by a variety of genes, disruption can lead to skin barrier diseases. Epidermis development. Shown are the intermediate stages of skin development and acquisition of postnatal keratin expression. Mesenchyme fibroblast and adipose development. The dermis originates from the mesodermal layer, also called mesenchyme. Main supportive cell, fibroblast, though they look similar under histology, they are functionally diverse. Fibroblasts secrete collagen and extracellular matrix, but also regulate keratinocyte function and skin homeostasis. Fibroblast development. Regional origins. Ventral body fibroblast from lateral plate mesoderm. Head fibroblast from neural crest precursors. Back skin fibroblast from somites, dermatomyotome. Fibroblasts maintain the ability to transdifferentiate, retaining their mesodermal flexibility. Adipose development. Adipocytes come from lower dermal fibroblast progenitors. Functions: energy balance, immune surveillance, hair cycle regulation. Types: white adipose tissue, WAT stores energy as lipid. Brown adipose tissue, BAT burns energy to generate heat. Found in paracervical, interscapular, supraclavicular subcutaneous regions. Melanocyte development. Melanocytes arise from neural crest cells. Present in the epidermis by 8 weeks EGA. More abundant in the dermis during development than adulthood. Persist in some dermal sites postnatally: hands, feet, scalp, sacrum and buttocks, common sites of blue nevi. Clinical correlates: piebaldism, defective melanocyte migration, localized albinism patches. Development of the cutaneous nerves. The skin contains a complex sensory network responsible for touch, temperature, pain, and pressure. Correct placement during development is carefully orchestrated. Types of sensory nerves. Classified by sensation, touch, pressure, temperature. Location. Deep dermal sensors, Pacinian corpuscles for pressure. Hair-associated sensors, light touch. Myelination. A-beta fibers, heavily myelinated, fast conduction, touch and pressure. A-delta fibers, thinly myelinated, sharp pain, temperature. C fibers, unmyelinated, dull pain, warmth, itch. Developmental origins. Sensory neurons develop from trigeminal ganglia, head innervation. Dorsal root ganglia, trunk and limb innervation. Development begins after motor neurons are established. Somatosensory nerves extend toward the skin periphery, guided by signals, likely from keratinocytes. Merkel cells. Specialized epidermal cells that find touch sensation. Derived from basal keratinocytes. Appear as early as 12 weeks estimated gestational age. Found in high density at volar surfaces, palms and soles, appendages and nerve-rich areas. Adnexal development. Adnexal structures are the skin appendages, hair, sebaceous glands, sweat glands, nails, apocrine glands, dermatoglyphics, fingerprints. Their development shares similarities with limb appendages, teeth, and mammary glands. Central signaling molecule, beta-catenin, active in keratinocytes and fibroblasts. Hair development. 75 days EGA. Hair placoid forms in epidermis, thin, columnar keratinocytes. Condensed fibroblasts gather beneath. 80 to 100 days EGA. Hair germ undergo PEG stage. Epithelium wraps around fibroblasts, forming the dermal papillae. 130 days EGA. Follicle produces lanugo hair, first keratinized shaft. Lanugo is shed before birth. Bulge stem cell compartment. Maintains hair cycle. Appears as a bulge in the follicle wall. Hair follicle development. Shown are the stages of hair follicle during development, with a focus on the overlapping signaling pathways between initial hair follicle morphogenesis and postnatal hair cycling. Sebaceous glands. Develop during bulbous PEG phase, 13-14 weeks. Activity, high in embryogenesis and puberty, low in childhood. Usually associated with hair follicles. Variants, Meibomian, eye, Fordyce spots, mouth, Tyson glands, prepuce, Montgomery glands, areola. Nail development. Preliminary nail forms and is shed in utero, second trimester. Sweat glands. 12 to 13 weeks EGA, volar, palms and soles. 20 weeks EGA, rest of body. Development mimics placoid formation of hair follicles. Apocrine glands. Develop later in hormonally responsive areas, axillae, genitals. Dermatoglyphics, fingerprints. Begin 7 weeks EGA. Caused by volar pads, fibroblast accumulations in palms and soles. As volar pads involute, epidermal ridges form via basal layer hyperproliferation. The skin requires an extensive vascular network to support growth, barrier function, and thermoregulation. Development begins with angioblasts, the stem cells of the vascular system. The skin contains hematopoietic cells, mainly lymphocytes and myeloid cells, which play roles in immunity and surveillance. Their origins are linked to systemic hematopoiesis, not purely local development. Early development. The yolk sac generates early myeloid lineages. By 10 weeks gestation, Langerhans cells, specialized antigen-presenting cells of the skin, are established. Later waves. A second wave of hematopoietic development occurs in the dorsal aorta, followed by contributions from the fetal liver. T cells. Not all skin T cells are recirculating. In adults, naive precursors can differentiate into skin-specific T cells, but this does not occur in embryos. Mosaicism occurs when a novel mutation arises during embryonic development. Leads to a subset of mutated cells coexisting with normal cells. Clinical significance. Mutated clones expand along embryonic developmental pathways, producing characteristic patterns on the skin. Classic pattern. Blaschko's lines. Appear as linear or curved streaks, reflecting the migration and expansion of embryonic clones. Seen in many cutaneous genetic disorders. Conclusions. Careful understanding of skin development, a first step toward harnessing cellular therapy. Still large gaps in knowledge relevant to this and other avenues.